So today we're starting the next chapter. The next chapter is chapter 11, which has really, if I was writing the book, I probably would just continue on lipids because it's about lipids. It's just a very more specific portion of it. There is one available already online for you to use to take notes. Okay. Which, let me read the real one. Here. Let's enter him. No, I'll just use this one. All right. So now we're specifically going to be looking at the lipids with respect to the biological membranes and how they're used for transporting. And we'll also talk at least a little bit about, towards the end of this chapter, and in the special for that matter, a little bit about some of the specialty characteristics or special uses of membranes. For those in neurochemistry, we've actually already covered some of those before. Okay. Not all of it, some of it. This is, most of this right here, you actually already know, just putting it all together. The fact that for the membranes that are used in vivo are bilayers, you should already know that one. And spontaneously, they're going to form these bilayers. Now, it is true, and I don't think your book should talk about it, there's something called the micellar concentration. And we can talk about it briefly. Which just means that if you go to a low enough concentration of your lipid, instead of forming a bilayer, it'll form a micelle, just a single layer. It's called a micelle. Okay. And it's a property of each lipid. But for those that are used for cells, and we're talking about a lipid bilayer. Okay. And it should make sense they have a hydrophobic tail, or tails, technically, and then they're going to have what typically is the polar head groups. So the polar head groups are going to be in contact with the aqueous, the polar environment. The tails are going to be on the interior. It's kind of hard to tell in this picture, but if you look on the PowerPoint, the watermark used for all these has to do with that interior. It's a really cool picture, since it's basically a diagram that clearly demonstrates what a bilayer looks like. It should go without saying, but I'll say it, that the number one force caused by far, that causes lipids to form is hydrophobic. Okay, and it wants to exclude the water from the interior of the lipid bilayer. The relative rigidity or fluidity, those are going to be inversely proportional, has to do with the property of what fatty acids. Okay. And we'll also get to a little bit about the effects of stuff like cholesterol and proteins and things like that. Lipid rafts can also play a part into their fluidity and rigidity. And it should also be noted that you can have localized regions that are more fluid or more rigid, and those are almost always very, very important. It's not like the cell is uniform throughout. Okay, what happened there? All right, so now let's look at some of the pretty pictures. Let's look at bilayer. Does anyone know what that means by that plus, minus, plus? What's that supposed to show you? It's indicative. That came from the old book, I think, the Thompson book. What are they trying to show you there with the pluses and minuses? Don't overthink it. Charge. That's the part that's polar charge. And then they've got the interior that's hydrophobic. And the fact that it forms these nice cells that can form much more pretty pictures than what I could ever draw. Okay. As I alluded to just a moment ago, let me make this a little smaller. If you go to a low enough concentration, you can form what's called a micelle, where it still has a hydrophobic interior, but the hydrophilic exterior. And we actually use this all the time for detergent. Okay, that's what we utilize this all the time. Detergent, the reason why it works, usually they have, in the old days they actually used phosphates, but now we don't use phosphate detergent since it's bad for the environment. We typically use those that are sulfate based or polyamine based, something like that. But what happens is you want to get that grease spot or the oil out of your clothes, you use a detergent, they form these micelles to where the hydrophobic tails will interact with the hydrophobic oil on your clothes, ideally, and the hydrophilic head groups of the detergent will react with the water in the washing machine, so it just washes it right on out. Okay. However, in vivo, those that typically are more important are those bilayers. And then they use this term called a vesicle, which can just be a small bilayer, small spherical bilayer, and it's going to have an aqueous hydrophilic interior, the hydrophobic layer, of course, itself, and then it's going to be aqueous on the outside. Now, I say vesicle, you have to realize that if you're in Canada or you speak the Queen's English, they don't say vesicle, they call it a vesicle. Like you say vehicle, they say vesicle. Just let you know. FYI. Okay. Sometimes you can see this on a micrograph, like an electron microscope. So here we can clearly see the lipid head groups, the aqueous environment, and then we have the hydrophobic, relatively non-dense intermembrane region. Okay, so now let's start talking about the biological membranes and the effects of cholesterol. That's what the blink's on there, fill in the blank. Cholesterol in the animal membranes. Give everybody a moment to get across. This is the steroid. This is what I would expect you to recognize this and be able to draw the A, B, C, and D rings. The actual identity, like where the double bonds are and stuff like that, can change, but they always have those tetracircular, I think that's the right word. It has 4 circles, okay, 4 cyclic groups, and it always goes in this order. So we have cholesterol. Then I've just given examples of both a sphingolipid and a phospholipid. All 3 of these can be used to make up membranes. Okay. So what are going to be the effects of having cholesterol in the membrane? What's one of the effects? You look at it versus the other 2. More stable. More rigid. For what? Well, I mean, no. Which is more rigid, the cholesterol one or those? The cholesterol is more rigid. Because it's kind of hard to tell here, but first of all, it is hydrophobic, but secondly, it is relatively thinner. It's not a real true thinner, but almost like, to me, it kind of looks like a corrugated cardboard look. And it has that really, really tiny polar head group. When we're talking about the cholesterol that's usually used for the membrane, whenever we're talking about cholesterol for storage, remember, those are usually cholesterol esters. So it's even less polar because the alcohol gets replaced by an ester, by an acyl group. You can also see, especially if you look at phosphatidylcholine, it's really easy to see. It looks like a person. I've never done this before. It's like, or something. But you can clearly see where it's got the head group, and then how it would increase through the fluidity or decrease the rigidity because of the fact that you can't have it stacking. Okay, the same thing is true with the sphingomyelin. It's a little bit more difficult to tell in that picture right there. Okay. So let's bring it on home. So it increases rigidity. It is a characteristic of animal rather than plant membranes. Okay. So therefore, I could have asked you which is more rigid or more fluid. Animals more rigid or less fluid. Plants are more fluid, less rigid. Just typically, we're not talking about cell wall. We're talking about the plasma membrane. Okay, has nothing to do with cell wall. Now, one thing to note are that prokaryotes usually contain no appreciable amounts of sterols. Okay, so they are going to be the most fluid of the 4. Once again, we're not talking about a cell wall. We're talking about the inner plasma membrane. All right. Check something out. That is freaked. It's freaking. All right. Any questions so far? This is one of the reasons why typically if you eat a meat-heavy diet, your exogenous cholesterol levels will be much higher versus your endogenous. What do I mean by exogenous? fancy word for consumed exogens from out, from outside. And endogenous would be cholesterol that is made by your own body. Okay. Western diet typically is much higher in exogenous cholesterol, stereotypically speaking, than other diets. Okay. I forgot what I blatantly plagiarized this from. I should say plagiarized. Blatantly taken this from. It used to sit in the bottom. It's from a bed. I know that much. I can't remember, but I got that from they were advertising some bed. But years ago. This was really cool. It has absolutely nothing to do with lipids. But I forgot even how I found it. Like Google images, and I just put in there, temperature or something. And it popped up, so. All right. So what are the effects of temperature on membranes? What happens if you increase the temperature of a membrane? Well, just as I say, we're slowly increasing. Yeah, you go high enough. Okay. As you slowly increase it, what's going to happen? Is it going to become more rigid or more fluid? More fluid. See, it's logical. See, I don't need to teach you guys this. You already know it.
So would certain parts of the body have more cholesterol to counteract that?
Ego. You jumped the gun there. Oh, that's something. Not guns. That just reminds me of something else. I haven't checked on it. But yes, it does. It does affect, or it can affect, not just temperature, but there are other reasons why you want to have differences in rigidity. Okay. Which brings us right. You think that could have been explained out there? Okay. And I don't. So why is that not coming up right? I apologize if that one looks really weird. So what biomolecules make up the membrane? Comprise means fancy way of saying make up. There are proteins. What else are there? There are glycosides. Usually we're talking about peripherally, but yes, the sugars. What else is in there?
Phospholipids.
Thank you, there are lipids. Thank you. Okay. Nucleic acids, not really. Okay. That's the one that we don't have to worry about. Doesn't matter what the identity is of the proteins, the lipids, the types of lipids, and stuff like that for the membrane. Yes or no? Yes. Okay. Why? Are all membranes created equal? No. Unlike man, all membranes, and women, all membranes are not created equal. Okay? So not only are they not created equal, so you can either do it based off a body part or tissue, but you can also do it based off a part of the cell. So what's one region? See, unfortunately, using this, I can't get it to. Let's see if I can type it in. There's a part coming up that I usually show you. You can do it. Let's see if I can just pull up the. Of course, that's a mouthful. I will try to type it in. Okay. I'll show you that not all membranes are created equal. What's just in general? What do you suppose? Do all membranes have the same type or the same amount of proteins in them? No. There are some membranes that will have more or less. What about the same type of lipid? Do all membranes, are they all composed of the same type of lipid? In relative concentrations? No. Okay. There are changes, there are differences. Okay. That brings us to this whole idea of the fluid mosaic model. This is something you should have probably learned all the way back into biology 1 or 2. I would assume you'd at least touch upon it in cell molecular biology, since it's like a tenet of it. Of course, it may be in the fact that it's something that people also just assume you already know, fancy term. But what is the fluid mosaic model? If you break it down.
Membrane is constantly shifting.
Okay, so fluidity would imply, I think that's what you're going with the shifting.
Movement.
Like there's movement. Okay. What's a mosaic?
Containing multiple parts or.
Well, of course, what is it? Where do we get that term from? What is a mosaic? If we weren't talking about the fluid mosaic model here, what is a mosaic? Where do you see mosaics? There's one possible place to see mosaics. An art museum. Right. So what is a piece of mosaic art? Bunch of little broken pieces, bunch of little bits and pieces, little bits and parts are supposed to all have their roles in it and everything else. Well, that's what they use to describe the lipids, the biological membranes. So fluid, you do have lateral motion. They're not static. Okay. And so I forgot where one time I used to have it. They talk about like the proteins floating on the sea of lipids. It's very poetic. It's a great way of saying things. But you can have movement this way. We'll get later on whether or not you can have movement transversely, but at least laterally. Okay. Then there's mosaic, and that's the fact that they are separate entities. Now they're not quite mutually exclusive. We'll get to what that means in a moment. And one other thing, kind of put down here. And the reason why, unfortunately, since this doesn't treat it as a PowerPoint, I have this link to a website where you can click on it and it gives this really nice website. So I think it works in the PowerPoint. Where it goes over lots of detail. It comes from CUNY, so the City University of New York, and so it's really nice. It's not really high class looking, so you can find it where the normal light. That's why I mean versus table. They do a really good job. But one thing I wanted to point out is that there's also a difference from the sides, from those that's extracellular side versus that side that's intracellular for that mosaic. Let me see if I can get. Let me see if I can get this website up on this one. Oh, I bet this is probably not even connected to the internet. Let's see. Browser. Got Brooklyn. .cuny. It's .edu, I think. .edu /. So on the Mac, there's /. Nope, wrong one. Frontward / biology. If I get all this done and typed in here and it's kicked me off of the internet, I'm not going to be happy. KBIO 4FV. Yes. / page. /PM. Which one's underscore? Is that underscore? Yes. Most.htm. Go. Yes. That's it. There we go. Hallelujah. Okay. So like I said, it's not the most glamorous looking here. But what I liked about it is they actually divide it up. And I would contend not be completely surprised if I were you to see something along this term in order that you need to be able to rationalize why this is true. So they're showing a little picture that's, really ugly. But if you look at their tables, that will show you the amount of proteins, lipids, all that kind of jazz. No, don't do that. Here we go. Good. So if we just look at this part, oh wow, that even did work. If we just look here as a membrane in general, and we look at the percent protein, percent lipid, and percent carbohydrate in this top, and we're going to do this next one in a moment. We see myelin, we have the erythrocyte, which what is an erythrocyte? What? Okay. We have a blood cell, we have the mitochondrial, and these are just random things, the mitochondrial inner membrane, and then an amino. Okay, so you don't do, I'm going to talk about the microbiology here with respect to the amoeba. But if we look at these three up here, if we notice, which of the three is going to have has the highest protein concentration? Mitochondrial inner membrane. And it has no sugar at all on it. Why does that make sense? Whereas if you look at the blood cell, it has the highest concentration of sugar on it. Why does that make sense? Why it has to be, why? So let's first actually let's look at carbohydrate. Why does it make sense if there's going to be more carbohydrate on the blood cell versus myelin? It's what? Recognition? Like what's an example of that with respect to recognition?
Blood typing.
Blood typing, and what was that called? So what was that called in this last chapter? Someone said it. Ganglioside. That's a ganglioside, which is made of carbohydrates. So yes, so that's what the blood type. This is one example. So we're talking about recognition. Why is there the highest concentration of lipids on violet, not violet? What is myelin? What is it used for? Insulation for neurons. Okay, covering the neurons. So why would it have the highest concentration of carbohydrates? Not carbohydrates, of the lipids. Or you can think of this, why would it have a much lower concentration of protein, the lowest of those three at least that we show here? What else has to occur? What?
Could it be used for energy?
Energy? Close. I mean, in chemistry, in general chemistry, we make that argument, but probably in biochemistry we would call it energy. What is it helping to do? Propagate. The electron potential of an electron, you can imagine propagate the signal of the neuron. So why does it make sense that if you had all of a sudden you put lots of proteins right there in that myelin sheath, what would it do to it? It would at least slow it down. You make that little speed bumps instead of if you look at the nodes of reindeer, there's like this in between, but also you got these little jagged looking pieces of protein and stuff. Now it comes to the mitochondrial inner membrane, which actually doesn't look, I think that's also in the bottom. No, I lied. Okay, so why we come to the mitochondrial inner membrane? It's almost 80% protein, which is astronomically high. Why does that make sense? First of all, it has no sugar. And secondly, why is it 80% protein? What is happening there?
Transport. Protons getting shuttled across the membrane.
Okay.
And oxidative phosphorylation.
Here we go, oxidative phosphorylation, and they don't call it the proton pump, but what's connected with oxidative phosphorylation we cover next semester? Someone said it, I thought. It is a type of transport. Not only does it transport protons, but it transports electrons, electron transport chain. Thank you. We're trying to put together. Electron transport chain and oxide phosphorylation. They call it the ETC oxphos. Technically, there are two, but there are a couple things that's occurring there. It's all protein based. So that's what that inner membrane is primarily used for. And there are also other things that do, like the transport of things. I call it the holy of holies because it's kind of hard sometimes to make the integration. But I call the mitochondrial matrix the holy of holies. And so it's very, very selective on what gets in. So you have to have a lot of other transporters too, and shuttles and ways to get things across that membrane as well. But it's very, very high. I'm not going to touch amoeba. I'm not an amoeba file, so. All right. So then we can also, not only that, we can even break down the actual cell. We've already covered myelin. You can also see there's a difference in the type of of lipid within the cells. So we have like the rough ER, we have the nuclear membrane, we have the plasma membrane, and then we have, well, for E. coli, it's that plasma membrane. But once again, I would expect you to be able to use some rationale and some logic to talk about why there would be differences between some of these. Like what is happening, at the rough ER that would cause it to have a higher propensity for certain types of phospholipids versus particular why the plasma membrane, for example, has phosphatidylserine, has the highest concentration of the group, has the highest concentration of phosphatidylserine. I may not know the phosphatidylserine yet. Hopefully you will by the end of this chapter. Okay, that's it for right now. Pick up right now where we left off. I like this figure. It came from, it's not from the current edition of the book. I'm assuming that they would have it in there because it shows all the different varieties of components that are available for a typical membrane, maybe not typical membrane, for a membrane. One exception, I just want to point out one thing, is what you will see here is they call this a peripheral protein covalently linked to a lipid. If you put that, you'd probably only get partial credit because technically speaking, what's the difference between an integral protein and a peripheral protein? What? What is a peripheral protein? It's on the surface of one way or the other, and so it's affiliated with the membrane. Usually we're talking about ionic interactions, typically, or protein-protein interactions. Okay. Now, the same thing goes with this one over here. That one right there is that's not a peripheral protein at all. Okay. Even in your book when you read about it in a moment, when you see it, you're going to see the reason why. Integral proteins means that they have to have some type of integration into the membrane. They do not have to completely traverse the membrane to be considered an integral protein. And so this one, like when we talk about the types of integral proteins, there's a whole class of integral proteins that are covalently linked to lipids. So that's why I don't know why on this figure here they put that. And this one I'm assuming is just a typo because that should be integrated with phospholipid for it to be a normal peripheral protein. That's the only caveat that I put on this. But we can have integral proteins that do completely transverse the membrane, but they do not have to completely transverse the membrane. And then we can also have different components be modified either with protein-protein interactions or glycosylation, some type of sugar group added on, or even some type of lipid groups that are added on as well. Okay. So this is just to carry on with that, and I almost put a question about this one in particular on the thing that's due on Monday. Don't turn it in. Okay. But we can see how the composition of the lipid and lipid identity does vary even within the same cell based off of what organelle and what membrane it's on. Okay. I could even throw up like a trick question and ask about like Cajal bodies or nucleolus. Why would the nucleolus or Cajal body be a trick question? What? They don't have membranes, so they're okay. So, yes, you get the metaphorical cookie award. Yay. All right. So actually, we've already talked about this and the whole idea that they're peripheral, integral, and there are those that are deemed amphitrophic, meaning they're found in both. Okay. I would expect you to be able to talk about the different ways to isolate these proteins. Okay. So and this should make sense, since a peripheral protein is primarily going to be bound with an electrostatic interaction. Okay, usually, yes, there are some that may be protein-protein interactions, but even then, usually we're talking about electrostatics, which is just the fancy term for ionic, right? So what you can do is if you increase the ionic strength of your solution, if you're trying to do protein purification, you can actually compete it off. It'll fall off. Whenever you get to that PI of sorts, of that ionic strength, there you're going to be able to knock it off. Whereas an integral protein is not going to work that way because they're going to be somehow bound to the interior of the membrane. So you're going to have to do some type of detergent treatment usually. What is ultrasonification? What are you using there if you do ultrasonification? Sound waves. It's so much fun. We actually had this on the heater. So which I've used here. I bought it a few years ago. We've been able to use it for doing protein purifications. But yes, and so it looks like a little needle, little probe, and it sticks in and it vibrates at such high frequency that when it's a sound, the sound wave will break up the membranes. Okay. Typically, you still put some type of detergent in, even a low level detergent or ions in there. Usually, this does denature. Many times, if you're careful with the peripheral protein, you won't denature it. Now, if you get to really, really high ionic strength, it may precipitate out, but usually you can play around with it and not denature it. But almost always they're going to denature this. And we'll talk about the reasons why. Actually, I can talk about the reason right now. I can draw it. So we can imagine, and don't hate me for my drawing ability. So here's a lipid, the lipid bilayer. And so here's the integral protein. Okay, that's my example. So whenever you put in something to a detergent, it's got to compete with this portion, but in order to do that, it's going to be removing this. And so we're going to see that the detergent comes in and competes off the lipids. But in doing so, it's not going to carefully extricate that protein and then have, the detergent around it. No, what typically happens is it's going to unfold that protein, and then the detergent will just stick to the parts of the protein that are hydrophobic, or even completely encase it in a micelle. Okay. So it'll unwrap it. Okay, then the term amphitropic, of course, amphi means both. And what happens there is usually, many times, they're covalently bound via a lipid anchor, or sometimes it's some other type of modification that's going to cause it to want to stay kind of half with the membrane, half without. And this is what's really important, is the fact that many times these proteins, whether or not they're peripheral versus integral, is going to be dictated by some type of regulatory process. And so some of the really important ones that we'll talk about a lot, especially next semester, are farnesylation, which is a type of lipid. Okay, like RAS protein, we talked a little bit about RAS, I think, so far, but RAS, which is an oncogene, or it can be oncogenic, I should say. It has to be farnesylated in order for it to associate with the membrane and for it to be active. And also phosphorylation. Okay. Does anyone know what's palmitoylation? What's the palmitoyl group? Goes back to organic chemistry. The olation just means that, it's attached. So the palmito would be what kind of functional group? It's a common name. It's a fatty acid. It's one of the specific fatty acids. But it's been fatty esylated. Okay. Remember, whenever they would do naming carboxylic acids, they drop the ic acid and then they change it, so that's what they've done there. Palmitoic acid becomes palmitoylation, or a palmitoyl group. Yes, we are here. We made it. Most of us made it. All right, so this is just to show. It's almost as good as my drawing. Almost. Okay, but we can see the peripheral protein that's up here at the top. It's peripheral due to protein-protein interactions, but we could imagine having one that is just associated due to an ionic interaction with the phospholipid membrane itself. Okay? But you can compete that off by either changing the ionic strength. You can add in urea. Urea, though, typically will definitely denature it. Urea denatures proteins just in general. Carbonate, of course, is just an anion. Okay, this is an integral protein, and so it gets removed by the use of a detergent. So that's trying to show how, now they kept it all the same, but in reality, to pluck it out, you're usually going to have it fall apart. And it's also usually more difficult to try to remove the detergent off of it. Like if you were wanting to study this protein, they're really, really difficult to work with. Okay, and then we have one that's an amphitrophic, that's just based off of some type of biological regulation. We have one here that's actually, we're going to see it's a specific type of integral protein due to a phospholipid anchor. And phospholipase C here, if you remember, it cleaves at a phospholipid right before the phosphate group of the phospholipid. And so it would then give off this protein. Okay. So, and I also apologize, for whatever reason, whenever it converted this into the PDF, I was able to fix it a lot on yours, I think, it's online. But it dropped, like, the first letter. It changed the font and dropped the first letter. It's supposed to be dictated. Okay, so these are the different types of integral proteins. I do expect you to know them. There are 6 types. And they matter based off of, or they're divided up based off of the directionality. They also are based off of the times they transverse the membrane, and whether or not they have lipid anchors. Okay. And also whether or not more than one protein all coming together for a big complex. Yeah, I'll give you a moment to get caught up. Okay. So, if you notice on this one, they have the sides of the membrane labeled. So this is a phospholipid bilayer, and so we have the extracellular side, which is on your right, and the cytosolic side on the left. So that's important because I have to know that in order for me to remember the difference between type 1 and type 2. For me, those are the ones that were the easiest to always get mixed up. Both type 1 and type 2 only transverse the membrane once. So then it's the direction that they transverse the membrane that's important. And so the way I remember it is I have to try to think of everything based off of the extracellular, the outside portion of the membrane. Because then amino comes first in alphabetical order, and then carboxyl comes last in alphabetical order. So amino is type 1, carboxyl is type 2 on the outside side, on the extracellular side. That's the only difference. For a type 3, this is a single polypeptide that transverses it multiple times. It doesn't matter the directionality here. Type 4, whoa, don't do that. Type 4 is different because this is one that we've actually, no, I take that back. We haven't talked about yet. No, I was going to say, we have in neurochemistry. So neurochemistry, those who are in neurochemistry, we talked about the receptor tyrosine kinases and how you have 2 proteins that have to come together in little graphs to get activated. That would be an example of a type 4 interaction. Okay. So what it is you have multiple proteins, multiple polypeptides that all come together and they form like a big complex. Type 5 is a type that was mislabeled on that one figure because it's a protein that's got a lipid anchor on it. And that's how it's held to the membrane. And then type 6 is a mixture. Okay? So it transverses the membrane and it contains lipid anchors. Okay, so are there any questions on this? Because then I kind of change. Yeah.
Does it matter how many, is it only specific to like say 5 times for type 3 and type 4, then it transfers 4 times?
No, you can transfer like 7 times.
Okay.
No, that's just happens to be the length. And just like type 4, you can have 2 polypeptides, or you can have 18, you know, things that are multiple polypeptides that come together to form one functional unit of something. Okay. This is not things, it's things. Whoops. I don't know why it did that. It annoys me. Okay. So kind of changing here a little bit. I mean, all of this thing has to do with integral proteins. Now we're going to start thinking about the properties of an integral protein and how they exist. We can wax philosophical right now. Okay. So, what kinds of amino acids make up an integral protein? So what we have to think about are the properties of the amino acids themselves. And here we're talking about the R groups, not the backbone, the R groups. Once again, this is a reminder, don't forget your amino acids, because they're on almost every quiz and exam and everything. Secondly, we have to think about the biophysical properties of the biological membranes, and then also we have to consider the location of these. Okay. So, I'm going to just use this as a little. Let's say that we've got a protein right here. It's an integral protein. There's a part of it that's out here on the outside. It transverses. That's a really crappy looking alpha helix, I guess. And then it comes back out. Something like that. You can make one of your own. Whatever. Now, it doesn't matter where we start. Oh, I didn't. Actually, let me redo all this. That way we can practice. I'll try to make it a little bit more. Okay. That's a farnesyl group. My version of farnesyl group there. So what class or what type of integral protein is this one?
Type 6?
It's type 6 because it transverses and it has a lipid, since I told you that that was supposed to be a farnesyl group with lipid on there, or some type of anchor on it. Okay, so it's type 6. All right. Now, and it doesn't matter to me where we start. We can start up here, we can start down there, we can start in the middle. It doesn't really matter. Where would you like to start? And we'll say that the top part is the extracellular space, the bottom space down here cytosol, but you have to realize that it could be right the opposite of all. Where would you like to start? Josh? Extracellular. Awesome. Okay, and then just to review once again, I was saying that this is the extracellular part. This is cytosolic. This is obviously the middle will be inside the membrane. And it's kind of hard to tell here, but this is the, whoa, I did not mean to do that. This right here is, I can see it, can you see? Those are the phospholipids, like that's the head groups. So these would be the fatty acid portions, and then this would be the other head groups. So if we start here at the extracellular space, what kind of amino acids do you suppose are going to be right out here? What type of environment is that out there? It's aqueous, okay? So what's some of the types, or what are the types, or what is the type of amino acid that you would expect to see a higher amount of right there? What's one amino acid that you can see right there? What? Maybe glycine.
Polar.
Polar. Why does it make sense it's going to have polar ones? Because water is polar and it's in the aqueous solution. So typically you're going to have polar ones. What else could you have out there? What else is another common, or what's another one that could possibly be interacting with the extracellular space? Charged ones. Those that are, you could either say those that are acidic or basic or negative or positive. It doesn't really matter so much. Okay? So those are the ones that we tend to see more of. Now, let's get a little closer. Let's say this right here is a phospholipid bilayer. Right? So we are specifically saying these are phospholipids. What amino acids would you expect to see a lot of right here in this general area? Be specific. If it's a phospholipid, what is the identity of a phospholipid typically? What do all phospholipids have in common? Phospho, the phosphate, and what's so special about the phospho or the phosphate part? It's charged, and what's its charge? Negative. So what do you suppose would be, what would be an ideal amino acid right around this area? Positive one, so what would be an example of that? Lysine, arginine, and histidine. Yeah. There's one other type that many times you'll see right at this interface that wouldn't make sense for you since I don't think you learned that in organic chemistry, and those are the aromatics. Sometimes you'd see like almost like phenylalanine or tyrosine like laying right alongside that. That's because once again it can kind of make a special type of aromatic interaction where there's the hole in the middle. I can't do that with my hand. I'm not about to shoot my hand. And so then you can imagine having the phosphate lipid that kind of sitting there, almost like a little hamburger bun kind of interaction. So don't be alarmed if you do see some of the like phenylalanine, tryptophan, and tyrosine right there at that interface. But right here, many times you do see those are positive charges. Now, if we keep moving on down, what are some of the amino acids that you would expect to see here within the membrane? The nonpolar. And this is another time that you do many times see alanine and glycine in some of those. Usually, like this is just a little asterisk, like rule of thumb, you have English, whatever. If you see, like if they are doing, we'll see a hydroxy index in just a moment. If you see a structure or something, and all of a sudden you see one or two charged amino acids or polar amino acids here inside what would be the membrane, they are very, very important. God put them there for a reason. And many times it's for protein-protein interactions. It's almost like a way to lock the two proteins together. As you can imagine, if you have one that's a positive charge right here and one that has a negative charge, they're going to really be attracted to each other because they're in a hydrophobic environment. And so it's almost like a way to Velcro them together. It's not really Velcro, but that yet. Okay. And then you're going to see the similar types of interactions on the cytosolic side that you saw on the extracellular side. One little caveat. Does anyone know what amino acid gets farnesylated the most? I don't want to give you bonus points right now if you need that.
What's farnesylation again?
Farnesylation is a very long lipid. It's cysteine. This one, cysteine typically gets farnesylated, but you can also have like palmitoylation and some of the other menoxylation and the cosylation and stuff that can happen through O's and N's. O, N, and S, so the amino groups, alcohols, and thiols are the ones that typically get modified. So, but farnesyl transferase the enzyme that puts the farnesyl group on, it prefers cysteines. I just got one thing about that worked on. Okay, so now let's put this in. This is actually, it's gonna drive me nuts. Example, not example. Okay, I don't know why. This is wrong. Okay, So this is glycophorin. It's a glycoprotein. It's like blood cell. It's not that important here, other than the fact that I could ask you one possible one, and you can maybe you'd make an argument on this one, what type of integral protein it is, because you'd say, well, it transverses once. It completely transverses once. This is kind of like 1.5, you know, so maybe you can make that argument, but so be it. Okay. So, it doesn't matter to me where we start once again. But actually, I'm going to start here because I can actually reach it with my hand. So, right here, notice, what do you notice in particular, right along here? Here's our membrane, our phospholipid membrane. What's one observation, Quentin, that you can make? What's an observation?
That's some change in the types of amino acids used.
Okay, and what type is used down here? What?
The polar.
Yeah, not only polar, but specifically what type?
I'm sorry, what's that?
What?
Positively charged.
These are positively charged. So we have arginine, lysine, lysine. Those are all positively charged. You can imagine them like being attracted and hanging out right there with those phospholipids. Okay. And then see here, you have that tyrosine. It's not like, oh, what's going on there? You know, but that's because it's making those special interactions as well. Okay. We'll get back to this in just a moment. So now we can see the kinds of amino acids that we have here. This is, you know, roughly alpha-helical. Any observations on this one? What kind do we see? They're non-polar. Yes, we have threonine. The threonine's branched too. Of the polar ones, it's not the most polar. But yeah, otherwise they are pretty much all polar. I mean non-polar. And then we do have the exception here in this little part that looks like an appendix or something. That's anatomy integration. But we have an appendage that looks like it's stuck in there. And then lo and behold, we do have these that are charged or polar. And you can see that they would presumably they're going to be interacting with each other. So, and I don't know glycophorin, what it does off the top of my head. But this must be very important for its structure. Okay, and so it's highly conserved. And then we have on the top, which I can't reach, once again, we have those that are charged. And then, since this is on an erythrocyte protein, it's a glycoprotein, you can even see the different sugars and the kinds of amino acids that have been glycosylated. So that's what that shows with the ovine versus the ending polysaccharides. Are there any questions over that? I'm trying to use that. So one method that they use is called the hydropathy index or hydropathy plot. That's supposed to say can, not an. It's can. Okay, these are really cool. So what they do is they take the primary amino acid sequence for a protein, and they have assigned how hydrophobic or how hydrophilic amino acids are. Okay, and it's got a value, and if you have a patch that's all together, then it can alter the value. And so this is a type of it's a bioinformatics tool that they use. And so a positive number means it's hydrophobic, negative number means it's hydrophilic. And so literally, if you look at glycophorin and you said, okay, there is no zero, but amino acid number 1, and they plot it, and so on all the way through to 128 or whatever the last number is on here. And lo and behold, we see one whole section that's very hydrophobic. So you can actually use these plots. Let's say that there's a protein that you're not for sure what it looks like, or whether or not it's an integral protein. You can get an idea of whether or not it is based off of this kind of information. Not only that, but you can also get an idea based off of this how many times it transverses. You get a minimum number of times that it transverses the membrane. So we know this has to transverse the membrane at least once, because we've got more than, when I took biophysics, they actually had like values. They put to this, I think it's more than, I can't remember now, 7 or so in a row. I mean, there is a value to put on it. That's not the important part. Okay. Does 7 is probably not right. I'd have to sit down and figure it out. I didn't have you guys memorize like rise per turn and all that kind of stuff. All right, so here's another. This one is bacteriorhodopsin. Its crystal structure is known, but let's say that we don't know it. For those of you who are not colorblind, it is color-coded. Well, it is color-coded for those who are colorblind too, but we just can't tell the colors. All right. So, just based off of this right here, it's a single polypeptide. So it's a single. What class of integral proteins is it? 1, 2, 3, 4, 5, 6, 6. Types. It's a type 3. Okay. Remember, type 1 and 2 only transverse at one time. Type 3, you have a single polypeptide chain, but it can transverse multiple times. Okay, so if we do the hydropathy plot for it, you can see there are those little loops and things that go outside. Yeah.
Based on the graph, how do we know that 6 and 7?
You can see where it transverses and it's not fully, it's like interfering with the head groups. It's not fully out there.
Yeah.
But no, I would, if I gave you that on a quiz or exam, and you told me 6, or you told me 7, and you justified it, well, the reason why is because it's, then I would accept either of those, as long as you justified it. They don't have the looping. Here, let me see. Let me do it on here because I can't reach that tall. I mean, this clearly has a loop. We can't see what's happening back there.
It looks like some of them, I guess, don't.
I can't tell the colors. This color going to this one has a little bit of a loop that you can see. Whoops. That one's got a big loop. That one's got a big loop. The one that we can't see what's happening down here, and the one that it is a loop. It's just not a loop that protrudes. A loop just means it's between two alpha helices or two beta strands. And so that's the determinants. And so we can see between these two right here, we have a really big loop. Not all loops are created.
I just couldn't tell if there was a dip for each one that protruded.
Not necessarily. Okay. Now, this is what it looks like. Once again, it's color coded. And based off of this, you can get an idea, once again, of a minimum number of times that it transverses the membrane, even if you didn't know the crystal structure, just based off of its primary structure. And so, where you can see 1, 2, 3, 4, 5, 6, 7 times. Yeah. That's my count voice. It's also, by the way, my Eeyore voice. I don't know why.
What are the dips indicate?
Pardon?
The dips indicate.
What do the dips indicate? It's a good question. So rather than me just giving you the answer, what do you suppose these dips? You're talking about this right here, right? Like, what does that indicate? If this is the hydrophobic region, so this would be.
Those would be the parts outside.
That's the part outside, and so that's why if we go back, that's why I tell you it would give you the minimum number. Like if I only gave you, if it wasn't color coded, I only gave you this and there were no numbers, I would expect you to probably say 6. There'd be a minimum of 6.
Yeah.
They know it's, well, first of all, they know the crystal structure, but they know that it's 6 and 7 also because, let me see if I can try to find it on here. Okay. Mm-hmm.
For some of them, they don't have that looping portion. Is that just
Because there's not between 6 and 7, and if you notice between 3 and 4, it's really, really close. Okay, so it's not a perfect science in that sense. Mm-hmm.
If on the exam we were given this, would you also accept 8 because it appears there's another peak there in the 7th loop, just as low as the one dividing 6 and 7? Yeah.
Yeah, if you said, okay, because chances are, if I were answering this question, I would number them, right? I'd say, okay, if that was my answer, there's 8 because I have 1, 2, 3, 4, 5, 6, 7, and 8 because of this, and now we're going to make that little note. Okay. Okay. All right, so these are just examples of different proteins that they actually know the crystal structures for. And it shows you all the charge residues. You can see how the charge residues typically are on in the aqueous environments. But at the same time, it does show the tryptophan's entire scene. That kind of also show you right where the interface are between the polar head groups and the natural lipids. And that's due to that kind of hydrophobic effect there. Okay. And I don't know what each one of those are. OMP-X, I think, is a bacterial protein. Multiporin is a pore. Potassium channel, phospholipase A. Of course, we talked about phospholipase A. Okay. Oops. That's supposed to say not all alpha helical. Can't you tell? Like trying to figure out what the text was when it transported it from one version to the other, and it lost. It's supposed to be not all alpha helical. Because in the previous ones, we've been showing alpha helical versions, but we can have those that are either alpha beta or those that are predominantly beta sheet. Okay. Usually, if you remember when we did protein structure, a beta sheet's not planar. It's got a slight tilt, natural. It's called a twist, natural twist to it. And so those that are completely or almost all completely beta sheet typically are huge because you have to have that natural twist all the way around. Okay, how are we doing on time? Oh, good. Then, that's going to just drive me nuts. At least this one you can kind of figure out what it was supposed to say. It's supposed to be types of lipid anchors. All right. So here's a whole host. And once you can kind of get an idea of the different types of amino acids, you can expect to see that do this. I'm going to start on I'll start left to right. Actually, no, I'm going to start with phenylation, which is nowhere near. So, because we had mentioned it, and see, that's why I was kind of drawing to make it look like this. Farnesylation. There's another type called geranyl-geranyllation. Remember, it comes from this idea of an isoprene. Isoprene has 5 carbons in it, and they alternate double single bonds whenever they form a blue bond. So it's a conjugated system, but it's not. You can't tell that here because it was conjugated before it was used as a building block by your body. And farnesyl means 15. There are 15 carbons that make a farnesyl group. Geranyl means 10, so geranyl-geranyl is 20. There's 20 carbons on it. It's called the geranyl-geranyl group, and they call it geranyl-geranyllation. It's a mouthful. If it's 15 carbons on it, it's called farnesylation.
So would 30 be fonso or geranyl?
I don't think that it has a name. I know. I don't know why they do it that way. They just do. And the enzyme that adds a farnesyl group is called a farnesyl transferase. So knowing that, what class of enzymes do you suppose adds a geranyl-geranyl group? Farnesyl transferase adds farnesyl groups. So therefore, what enzyme adds geranyl-geranyl groups? Geranyl-geranyl transferases, GGTases. Farnesyl transferase they abbreviate as FTase. But yeah. Okay, so that's why I just wanted to point that one out first since there had been a question about that. This is a palmitoyl group. Usually it's on a cysteine or a serine. And they're showing it on a cysteine right here, and you can see how it gets stuck in there. You can have a myristoyl group also. I wouldn't ask you that you know that it's on the amino terminal glycine so much. Then we have as I mentioned, the farnesyl group, which is usually on a cysteine. And this says that's on the C-terminal cysteine. However, that's an oversimplification. We'll get to it. Maybe we'll get to it next semester. There's a whole process, a metabolic process that adds those on. But they typically are near the C-terminus. Okay. Then we can have this huge GPI anchor. Okay. And GPI is for what it does is it has a glycerol. It's a glycerol backbone. So see, it's kind of hard to tell here, but there's one carbon with the alcohol, another carbon that had the alcohol, and the third carbon that had the alcohol. This is a phospholipid, and then it's an inositol, and then there's a whole bunch of different sugars that are added on. Okay. Okay. Ooh, we are near. Let's see how far we. No, I better go. Okay, I would say it'd be a good place to take a break, but we'll go just a little bit further because I want to make sure we get a little bit to this. Okay, now we talked about the different proteins that are involved, how they're modified. Now we're going to start talking about membrane dynamics, which is really cool. One of my all-time favorite enzyme names happens in this part of the chapter, and so I love it. This part, nothing new. You can imagine lateral diffusion just means it can go side to side in all directions on the in the membrane. It's on the same leaflet, so a lot of times they need to call these leaflets. It's really fast. Remember I've talked about the fact that there's like that sea of lipids and you can float around. Okay. Whereas if you try to do trans-bilayer movement, when you flip from the outside in or from the inner leaflet to the outside, it's much more difficult, okay? And last year I talked about flip-flopping. Flip-flopping is good because we had presidential election time, but this is at a point, and you had a post on it. Okay, why do you suppose it's going to require, if it's uncatalyzed, it takes a very long time. You would die, okay? It's just on the amount of time span, because there are times you have to make sure you get lipids from one side to the other. You're going to have to, okay? So they have to be enzyme catalyzed. Why is it so slow to go from outside in or inside out?
Hydrophobic forces.
Right, it's hydrophobic here, and so what's going to be the difficult part?
Polar head.
Getting that polar head, you're through. Okay, so you have to have enzymes. Okay. So, here we go. These are flipase and flopase, and it doesn't matter which one is which. Okay? Because, let's see, when did I talk about? Oh, well, wait, it's two more. I don't want to give too much away. Okay, flipases and flopases. Flipases are called p-type ATPases. Flopases are ABC transporters. P-type ATPase requires ATP. Okay, P-type is a type of proton. Usually we talk about proton pumps. So I don't know if any of you have taken any Nexium or Protonics or what's the other one that now you do count. Prilosec, things like that. Omeprazole, you're doing the generics. Those are P-type proton pump inhibitors. So sometimes you can get these really weird side effects, if you notice, and one of the reasons why is because it's also P-type. Okay. So flipases is a P-type ATPase, and it moves, this is phosphatidylethanolamine, this is the abbreviation for it. That's phosphatidylserine, the abbreviation for it, which was given earlier in the chapter. All right, and it goes from the outside in. Flopases just moves generic phospholipids from the cytosolic to the outer leaflet. All right, so they go from the outside, they go from the inside out. So that goes outside in, inside out. Okay. This one we're going to get to in a moment. They require the hydrolysis of ATP because they're going against concentration gradients typically. Another one that we have, which if you have flipase and flopase, the one that can go in either direction, my favorite one, scramblase, which someone thought one time I said scrambled eggs, but no, it's scramblase. So it goes towards equilibrium and it goes either direction. It does not require ATP. Okay. So flipase, outside in, flopase, inside out, scramblase, no ATP required. Okay. And then I want to put a lot of emphasis on flipase. We're going to talk about flipase for this last couple minutes. Lipase is so important because you're probably going to forget about this coming semester. This comes up again next semester when we talk about metabolic pathways. Lipases only move the amino phospholipids. Okay, so that's phosphatidylethanolamine, okay, and phosphatidylserine, that's amino acid, and it's going from the inside. From the outside to the inside. It helps to keep it asymmetric. This is very important because you need to keep the PS, the phosphatidylserine, from that outer leaflet because it is a signal for macrophage. There are macrophages that recognize PS. And so, if you get the PS to start to go on the outside, that cell will die. Okay, it's a form for apoptosis. So the flipase is important for the cell to remain viable and healthy because it's going to make sure to keep the phosphatidylserine and the phosphatidylethanolamine as well on the inside rather than having it pop out to the outside and causing it to trigger apoptosis, macrophagocytic apoptosis. Okay. Are there any questions on that? Okay.